says we're live. I think we're live. I'm just going to confirm when I see it on YouTube and make sure we have a chat window and we'll get going. Let's see.
So good at
It
this.
says we're live. I think we're live. Oops. Live. I'm just going to confirm. All right. Here we go. Hello, Mycophiles, Pikes Peak Mycological Society and guests. Welcome to the August meeting of the Pikes Peak Mycological Society, another installment on Zoom due to COVID-19 that we're all very aware of. We're really excited. We have a special guest today, Giuliana Furci, who's going to give a talk and in a minute. And before that, I just want to introduce other officers and board members of PPMS who are here with us today. So we have our treasurer, Beth Leek, and our newsletter co-editor, Jessica Langley, and our newsletter co-editor, Mercedes Whitman. Thank you guys for being here. I think usually we just want to start off with a couple announcements. First, this meeting is live on YouTube because hopefully you're watching it currently live on YouTube. And you can ask questions through the chat window. I'll make a little note here. I'll just type something here. And feel free to ask questions in the chat. I will be watching that and I can relay those questions in real time to Giuliana. Quick business stuff. You know, we've been scouting the season. We got some rain in early August and everyone got really excited. We had a couple forays and we are doing forays when the mushrooms are out. They were pretty fun, successful forays. You'll get emails about those. There's a sign up that we are asking people to use so we know numbers. And then once we know how many people are coming, we're able to figure out how many foray leaders we need to have to keep people in smaller groups. Where masks are required, we're staying 6 feet apart, but it's outside and it's been pretty successful and pretty fun for those who could attend. We're going to continue doing that as long as the season permits, but as you know, the Western Slope is on fire right now. It's been a serious drought for the past couple weeks. So it's all rain dependent. And Brian Barzee has been scouting all over Colorado. He went down to the edge of New Mexico. He was out for like 18 hours recently and found not a single mushroom. So until we feel like it's worth doing, we're not going to you know, organize anything, but we're paying attention, we're scouting. And so keep an eye on your email and on Facebook for announcements on that. Another announcement is I think we've had new members joining and some people not getting emails. I just want to mention that in the past we've had issues with our server, emails from pikespeakmike.org being kind of thought to be spam, sent to spam folders, or sometimes sent to your promotions folder if you have Gmail. So keep an eye on different folders if you're expecting an email. And if you have any questions, you can always contact us via Facebook or email info@pikespeakmike.org. And also remember, we're an all volunteer organization. And if you know you want to be a member and you want to go on a foray, don't wait till a foray is announced to sign up, because that doesn't give us much time to read your email, process the payment, and send you the welcome letter. So if you know, if you hear that rain's in the forecast and you've been thinking about being a member, join a little bit ahead of time. Give us some lead time on that so that we can get the information out. Beth, is there anything else on that note you want to add?
No, just if you're not a member yet, you can sign up on our website on Pikes Peak Mycological Society. I forget what the actual web name
I'll put it in the chat, pikespeakmike.org.
Yeah, and click on join and you can do everything through PayPal.
Cool, thanks, Beth. And then we just released a new newsletter edition. It should have been in your inboxes. For anyone who's a member, you get a password for our forays page and our newsletters page, and then you'll get a newsletter emailed to you. And it's a really awesome edition. Jessica or Mercedes, do you guys want to say anything about that? You're muted. You're muted. Classic.
There it is. Okay. Sorry. I'll just say that for this issue, there's an awesome spread of this artist, Phyllis Ma, and some of her artwork is featured in this issue. And then Mercedes has also written a really great description of a day that she volunteered with the Coalition for the Upper South Platte, which does mycoremediation. One of our members contributed something. Her name is Lee, and it's about the relationship between mushrooms and bees, which is awesome. And then, you know, some great 4A pictures. So check your inboxes, check your spam if you didn't find it. But it was sent out by editor at newsletter.pikespeak, sorry, editor at pikespeakmike.org.
Send us stuff. We want new voices. Please.
Yes.
Don't be shy.
No, classic. Muted. There's going to be one more edition this season. Is that correct? This year?
Yeah, we'll have one more issue coming out in a couple more months. So definitely, just to reiterate what Mercedes just said, if you are considering writing or you have awesome mushroom pictures that you want to share, things that you've been finding, or something you're curious about that you want to do more research on, we welcome contributions from members and non-members too.
Cool. Thanks, guys. All right. I think that's all the business that we have at the moment. We're going to have our next lecture is going to be on September 23rd. Let me confirm that. September 23rd should be the Wednesday, fourth Wednesday of the month. And we're going to have a lecture by a chef, Zachary Mazzi. He's going to give a lecture on September 23rd. That's a Wednesday at 6 o'clock again via YouTube Live. So put that on your calendars and more information will be coming soon about that. Okay, awesome. So without further ado, I'd like to introduce Giuliana and let Giuliana take it away. So Giuliana Furci is founder and president of the Fungi Foundation. She's been the greatest promoter of the study and protection of the fungi kingdom in recent decades in Chile. She managed to make Chile the first country in the world to include the fungi kingdom in its environmental impact assessment legislation through its incorporation into the general environmental law, where its incorporation into the national species inventory is also mandated among other obligations. She's the author of several books and field guides and collaborated as co-author on Kew Gardens' first report on the state of the world's fungi, which is really awesome. If you haven't seen that, I think you can see most of it online, and it's really cool. Giuliana is curator of the FFCL Fungarium, is an associate of Harvard University, a member of the International Mycology Association, the Latin American Mycology Association, the Mycological Society of America, and the International Society for Fungal Conservation. Among her recognitions are the 2013 President's Award from the International Society for Fungal Conservation in Turkey, and the 2018 Salzman Award from the Telluride Mushroom Festival. I was really blown away by Giuliana's keynote lecture last year at Telluride Mushroom Festival, and I'm really excited to have her speak with us here at PPMS. We had really big plans of having her here in person this year. We were going to do a field day, collection day. We will try our best to do that next year, but obviously because of COVID, we have to do everything remotely. But yes, we'll try to get her here next year and do that again. And Giuliana, thanks so much for joining us from Santiago. And I don't want to take up any more time because I'm really excited to see your talk.
Thank you very much, Ben, and Jessica, and Beth, and Mercedes. And yeah, I'll be there next year, if COVID allowing. So this is just the beginning. It's not an end. So thank you very much for inviting me. I have a very heartfelt relationship with PPMS. I love Brian Barzee very much. He's a great friend. And so big shout out to him, and also to several other club members who are really, really great. Today, I mean, it's really hard to choose what to share, but I wanted to make a contribution to the club by showing you that what you see matters. Every encounter each one of you has with a mushroom or a fungus is super important. The fact that you coincide with a mushroom doesn't mean anybody else coincides with the same mushroom, and it doesn't necessarily mean that anybody else has ever coincided with that species before in history. So it's really important to acknowledge to yourselves that the mushrooms you coincide with are important, they matter, and you have the possibility of following a procedure that I'm going to teach you today, and your encounter can matter for science. So I'm going to share my screen, and we're going to do a step-by-step. Okay, you'll let me know, Ben, if you see the screen? All good?
I see the screen.
Perfect. Okay. So, let me see if I can make the move. Okay, so the first thing we need to define is the goal of our collecting. Are we collecting to eat, or are we collecting to voucher a species for science, to document a find? The reason I chose this is from my experience, over 2 decades of experience working for the fungi and with people who collect. And sometimes I'll get an email with, 5 incredible photographs of a species that's very special. And, the first thing I'll ask is, can you send me the sample? And 99% of the time, the answer is, I didn't take a sample. And so therefore, you're left with great pictures of a species or a specimen that you think is new to science in many cases, or at least new maybe to that region or to a country, but with no way to verify that, no way to describe it, no way to make sure it's new, and no way to really make sure of the mushroom's identity, because we do identification either through microscopy, or through molecular analysis of the specimen. In both cases, you need the physical mushroom, fresh or dry. So this is why we're going through these steps. So if you define that your aim for collecting is to eat, we can go around, and cut the fungus, cut the mushroom, as shown in the specimen that's in the forefront of the photograph at the front. And as most of you probably do, when you're foraying for food, you will find a porcini, you cut it, you'll clean it in the field, make sure, brush off any soil and put it in your basket. But when you're collecting for science, the procedure is different. So if you're collecting for food, there are several rules for that collection to be done in a sustainable way. The mushroom exists to produce and to disperse spores. And the function of the spore is for the fungus to reproduce sexually. What does that mean? It means that the spores carry genetic material that it comes from one individual, but it has to meet another compatible spore from a different individual, and they both need to produce their primary hyphae. They will, quote unquote, mate or pair, and they will produce a mycelium. So what's happening there essentially is that there's genetic recombination. It's like us. We reproduce sexually in most cases. And what's happening there is that you're getting genes from both parents, okay? So why does that happen? It has to do with adaptation. So in the measure that you're recombining genetically, you're able to adapt to environmental and other conditions easier than if you're cloning, where you're all the same, always the same, and you don't really have a chance to modify any aspect of a being to respond to environmental changes. So it's important to let species reproduce sexually, and therefore don't collect all the mushrooms when they're very, very small. Let some reach maturity and sporulate, and hopefully harvest them after they've sporulated, okay? Another step for a sustainable harvest or a regenerative harvest of wild edible fungi is to use a knife if possible. Don't yank them out of the soil, because when you yank them out, you're disrupting the mycelium. You are normally, if you yank out, a porcini or a bolete, it will come out with, a good portion of soil attached to the mycelium at the base of the stipe. So we want to avoid that when we're harvesting for food, because we're harvesting lots and lots of mushrooms of the same species normally. So use a knife, cut them, and clean them in the field, right where you've harvested them. Make sure that all the remnants of any mycelium or any parts of the fungus that you don't want to eat remain there, so that there's a possibility of asexual reproduction, i.e. cloning. So you can clone a mushroom from any part of its body. Mushrooms don't have, and fungi don't have, real tissue. So if we use the word tissue, if I use it, it's easier to use it. I want to make sure that you know that they don't have real tissue like plants and animals do. If we did a cross section of a porcini and looked at the microscope, all we would see are hyphae, just as we see in the mycelium. And the only part that's different in the case of porcini are the pores, and in the case of a mushroom with lamellae, it's the lamellae. That's the only part that's substantially different. The rest is all hyphae, so linear microscopic filaments, okay, one cell wall thick. So we want to make sure that they remain in the field, everything we clean off a mushroom, so that those hyphae can reproduce, reestablish, okay? Carrying the mushrooms and the fungi in baskets is mandatory, I would say. It's fundamental when you're harvesting a lot of specimens to eat. The basket allows for spore dispersal. If you're putting them in plastic bags or closed containers, the spores will not be able to be dispersed. And therefore, the mushroom will have less chance of reproducing sexually, okay? Other suggestions for when harvesting sustainably for food is, of course, never collect a mushroom or any fungus that you don't know who it is. It's a very, very simple. If you're not 100% sure of the identity of a mushroom, don't eat it. If you're 100% sure that it's edible, only eat it. And if there's a little chance that it may not be edible, don't risk it. Never collect all of them. Never collect all the carpophores or sporums or all the mushrooms. Leave some to reproduce, and leave some for the rest, for whoever may come behind you and may also want to harvest. That's something important to do. And as I said before, carry them in a suitable way, not only to allow for spore dispersal, but also so that you don't lose your harvest. Because if you pile them one on top of the other, without aeration, you'll probably get home with a lovely, maybe not lovely actually, with a really stinky soup of, squished, mushrooms that you won't eat anyway. So make sure to treat them with care after they're harvested. It's super important to clean them before putting them in the basket if you don't want to be munching on soil. And if you're okay with munching on soil, then you can just skip that recommendation. Now, to get to the topic of this talk, we're going to talk about how to collect for science, so that people like myself who work in fungariums, who curate fungariums, can help you know for certain what fungus you encountered, whether it's well known, whether it's special that it was there, or even whether it's new to science. And believe me, in my experience, there are many species new to science that citizen scientists overlook. And it has to do with the fact that they don't value the magic of their coincidence, and they don't collect. So I recommend you all get out paper and pen. I know this will be up on YouTube for a while, but we're going to go through some steps that may be useful for you to take notes on. So if I'm walking, I'm walking around in the forest with my basket and all my collecting gear, and I see a mushroom or a fungus. Not all fungi are mushrooms, right? So it could be a puffball, it could be a bird's nest, an earth star, whichever the case. The first thing one does is to look at it. Don't touch it yet. Take your time, look at the mushroom, and start taking photographs of the specimen in its natural habitat. What does that mean? Hopefully from far away first, and get your frame closer and closer into the mushroom. So without touching it, without disturbing or trying to clean the shot, first take a photograph from far away from what you're seeing, and start moving in, get down on your knees or on your belly and take a photograph from the side. And most importantly, be sure to use a size reference. Now, why do I say this? In the case of this fungus, which is a beautiful Leotia, with this photograph, I have no chance. I have no reference of the size. But as soon as I put a size reference in, you realize the dimension. So this could be huge. It could be the size of a bolete, but really it's 5 centimeters tall. And unless you have a size reference, you'll never be able to know that. Now, I don't recommend using lens caps because there are different diameter lenses. In some cases, a coin is better. And this is a Chilean 100 peso coin. If I hadn't used that coin, I would have no way of knowing the size of this Montagnea arenaria. It's a desert fungus, but you realize it's very, very small. This is smaller than a quarter, US quarter. It's bigger. It's like a 5-cent coin, probably. So make sure to use a size reference that's standardized. I normally use my hand, but my mother said to me many years ago, she said, you do realize that, hands shrink with age. And I was like, oh, okay, make sure, I change that. And I started using coins and just lost so many that I'm sticking to the hand. But because you always carry it and you can't leave it in the field, but carry something if you can, even a ruler, and make sure that you get those field shots with a size reference. It's incredible how much you go back to these.
There are some mushroom knives that actually have rulers on them.
Exactly. There are specialized mushroom knives that have rulers on them, and there are field guides. So for example, I've written several field guides, and in my case, what I do is that I include a ruler, in the book. So make sure if you have a field guide, check if they have a ruler, or if your knife has a ruler as well. Now, with time and specialization, one evolves in how to use the size reference. And in my case, for many years, I used the system that you see here on the screen, which is a label that has a bar. That black bar is 2 centimeters wide and 3 millimeters tall, okay? And so I know I have a size reference on the label. The label also has the location, it has the number of a sample, and it has the date. It has my initials, GF, because I'm the person who's collecting. And I'll get back to this later in the presentation, but this is a more evolved way of doing scientific collecting when you're really not even looking for edibles at all, okay? The second step is to gather the data. the data are super important. As I mentioned, when you're collecting systematically, it's important that every sample has its own number or name. So if I collect, an agaricus, the first agaricus, I will say 01, GF, so Giuliana Furci 01. Maybe I'll do 2020. The second agaricus I encounter will be 02. Even if it's the same species and it's in a different place, it will be 02. Never repeat the number. And it's very important because once you've dried specimens, sometimes they all look really alike. And unless that number is there, you have no way of relating the dried sample to the field photographs or to anything you've taken. So the best thing to do is to designate a name or number, make sure that that's in the photograph as well, with the size reference and the number. If you can, most phones have them, get a GPS coordinate. If you don't have, just get a coordinate, a location coordinate. Write down the location. So it could be, South Slope, Colorado Springs, 5 meters from a gas station. It can be like that. Just, something that will get you back to where you found the mushroom. Write down what it's growing on: soil, wood, burnt wood, tree, standing live tree trunk, standing dead tree, stump, and anything you can, anything that you see of that substrate that you can write down. It's important to write down what vegetation and what trees are around your find. So if you can identify that there's an aspen or that there's, a birch tree or there's an oak, write it down. If you realize that it's in a habitat where it's full of trees, there's mostly this tree and I don't know what it is, grab a leaf, put it inside your notebook, stick it next to the number of the mushroom you found. Smell the mushroom without touching it yet. Still, we haven't touched it. Get on your knees, smell. Most people smell, just by taking one, as we say in England, a whiff. Like, okay, I recommend smelling like a dog. So go, you get a better sense, do a little dog sniff on the mushroom. No judgment when in the field. If you see somebody sniffing like a dog, don't say anything. It really helps a lot, and write down what smells you might appreciate. The texture, is it fragile? Is it sturdy? What can you see, without touching it yet? And is it fragile? does it look firm? In mycology, poetry is not only allowed, it's desired. So, if it's slippery but smooth and firm, and or anything you can appreciate. There are times when one encounters a mushroom and one says, it's elegant. It's elegant, and everybody will see it, will say, oh wow, that's elegant. So write down elegant, and any word that comes to mind immediately. Does it have any stains on it? Is there something characteristic? That's important, and we'll get back to stains now. After writing all that down and getting these field photographs from, a wide angle, narrowing into, a macro shot, hopefully, we proceed to collect the mushroom. This is a blast from the past. I'm in this photo, I'm collecting a giant puffball in Patagonia, in Torres del Paine, in the year 2001, believe it or not. It's either 2000 or 2001. So that's a big blast from the past. It was so windy that I had to cover up my basket because the wind would blow the mushrooms out. So if you realize there's duct tape, you know, and like just a piece of plastic on top of the basket to prevent all my specimens from being blown away and all the work lost. So sometimes one can use a little spade, depending on the habitat. I think it's always useful to have a little spade with you when you're collecting. A knife, a hand lens, as many of you know. There are different types of hand lenses, but it's always good to have a hand lens with you. A good field knife. To collect, you can either use wax paper bags, you know, aluminum foil, or a tackle box. For scientific collection, tackle boxes are really useful because they have the compartments that prevent the samples from mixing together. And also you can, you know, put a few leaves inside and make a little moss or leaf bed for your sample inside the compartments of the tackle box. And there are different tackle boxes. The best ones in my experience are the ones that have removable separators. So you can adapt the size of the compartment depending on what, you know, fungi you find. So it's always good to have some of these with you, and you can put them in the basket as well. But when we're collecting for science, we're not as worried about spore dispersal as we are when we're harvesting for food, because when we're harvesting for food, we're collecting most of what we see of that edible species. When we're collecting for science, we'll see in shortly, the criteria for the number of collections or the number of specimens you collect is different. So with a spade or with a knife, gently unearth the fungus. We will not cut, okay? When we're collecting for science, we have to make sure that we get the base of the mushroom with the collection. We will not clean anything in the field, okay? So you unearth the specimen. Sometimes, I mean, I personally use my knife mostly if I'm not carrying a spade. So you'll be careful to not go, you know, horizontally into the soil, which will probably, you know, cut your stipe in two. It always happens. But, you know, go in vertically and try to, you know, slowly move around to loosen the whole sporum, the whole mushroom or fruit body. I don't like saying fruit body because fruits are for plants, but unearth it gently. And while you're doing so, are there new data that start appearing? Does it smell different? Does the smell come out? Does it automatically just like boom, collapse? You know, does it stay firm? Does it stain where I've touched it? Do insects fly out? You know, there are so many things that can happen. Make sure to write down or to register everything that's happening at the moment you unearth the specimen. When you were in the process of collecting a specimen, you can't always unearth them like in the photograph in the lower right-hand side, because they're in wood, like this, you know, the laughing Jim, the Gymnopolis that's on the right side, on the upper right side. And therefore, there's no chance to get to the base of the stipe when it's, you know, 30 centimeters in the middle of a tree trunk. So in that case, it's allowed, you're allowed to cut because you have no other choice. But once you do that, in the field, make sure to take photographs showing features, for example, how the gills or lamellae reach the stipe, which is super important in taxonomy to use field guides. One of the biggest features, macromorphological features, is how the lamellae or the gills reach the stipe. Do they touch the stipe? Do they touch the stipe and then run down, decur down? Do they not touch the stipe? Are they free? So for example, in the case of Amanita, all Amanitas have free lamellae. The lamellae don't touch the stipe, okay? Apart from the fact that they have white spores and other features. But there are some genera of fungi that with that feature will narrow down the possibilities of who it might be. And there are features like the margin of the pileus of the cap. So in the case of the Gymnopilus on the upper left, yeah, I don't know if I got them right or wrong before, but the Gymnopilus is the orange one. You can see that there are remnants of a veil or like a membrane on the edges of the cap. And those may be lost in the transport from the field to the home or to the cabin or to the tent or wherever you're working with your specimens. And that's why it's important to take these photographs in the field. And there are species that are very small and that even dry out before, just in the car ride to get back home. So it's important to take these photographs right there when you found them, okay? Many species change color when they dry out as well, and that's called their hygrophanous. So, as I mentioned before, how many? So when I'm foraging for food, I tend to want to get the most number of mushrooms or porcini. One has to be generous and one has to be sustainable and regenerative, and it's always important to leave some for the animals who eat them, leave some for the mushroom to sporulate and disperse, and leave some for whoever's coming behind you who wants to eat some as well. But when you're collecting for scientific reasons, you don't need to collect everything you see. And you want to collect what you photographed. So for example, if I'm in a patch of the forest, and I've seen this mushroom, and I did the whole process of photography that I just explained, but then I collect ones that are, 10 feet away, that's not recommendable. Collect the ones you've photographed that have the number on them, that have a size reference. And how many do I collect? Sometimes there aren't many. Mycologists normally say 1 is nothing, 2 is something, 3 is a collection, start of a collection. But most mycologists won't even stop to photograph or collect a species that there's only 1 specimen, unless you're certain it's really special or it's a big wood conch where you normally don't find many. So you'd want at least 3 for that to be a collection that will have enough material to do microscopy, molecular work, and to leave a voucher, for history. Because we mustn't forget that the reason we're collecting because we want to know who it is and we want to voucher our encounter with that species. And in a while, I'll show you what we do with what we voucher. But so if there are many, so, if it's like all Coprinus, little Coprinus, and there are, 20 of them, you can collect, 6 to dry, add on another 3 that you'll be using for the next step of cutting and photographing, and you can take some for a spore print. Okay, so in no case you need 20, but 1 is really insufficient unless it's a conch or, something very special. When you have specimens that are very, very small and you don't have a tackle box, it's important to put them in, either a wax paper bag or a little closed pouch from wax paper. But be careful when using, if you're going to use aluminum. I know that in the US you say aluminum, but we say aluminium. Aluminium paper, because the metal, when it's, how do you say, when it's folded up, can sometimes scratch and mark the mushrooms. Okay, so be careful with that. Collect, put them separately somewhere. You can make pouches from aluminium, you can make pouches from wax paper, and put them somewhere where you can keep them separate. And I very much like making moss or leaf beds with my specimens, and that's because the moisture of the leaves or of the moss keep the specimens fresh, so I have a bigger chance of seeing more features when I get back to a tent or a cabin. Now, the photograph I'm showing here is not, the reason it's here is that when people say, you know, use a moss bed, in my experience, a lot of people tend to do this that's in the photograph, and they'll just make, you know, one giant moss bed and put all the specimens there. But one trip in the field, you trip once, and there's no good field foray without at least one fall. They're all mixed, and most of them are broken, and the moss is everywhere. So as I said before, try to make the moss beds inside the tackle boxes, and just, you know, you can even cover the mushroom with a leaf and make sure that it remains, you know, moist. And then when you finally get back, you know, to your, sorry, one very important thing is the number you use to identify, the number or name that you use to identify your fungus in the field has to go into the bag or the tackle box with the specimen. Never separate the number or the label from the specimen that it belongs to, because it's very hard later to match which was which. So you get back home and you have to move on to the next step, which is taking technical photographs. And many of you may see photographs like these in field guides or in, you know, scientific papers, or, you know, posts, or you know, these sites like Mushroom Observer or iNaturalist or others. So the important thing is to have a monochromatic background. What does that mean? One color, ideally that it doesn't, not shiny, can't be shiny, because otherwise you just get shine of your camera or the flash or whatever you need to use. And place the mushroom on that, you know, I normally use fabric. So a non-glossy fabric, either gray or black. And you know, you put a ruler down, the same label you had. If the label got wet in the rain or from the mushroom, write it out again, exactly the same. And put it next to the mushroom, and take pictures from below, from above, get details of the lamellae. Do a section, a longitudinal section. So here's another example, you know, one way, the other way, you know, you get details of the annulus, of the margin. Do a section, a longitudinal section. I'm sorry, I can't say the word, like in this case, because here what we are seeing very clearly are characteristics that are important to ID the specimen. Here we very clearly see that in this case, for example, the gills or lamellae don't touch the stite. The stite is hollow. It has a hollow canal inside. We will be able to see the type of bulbous base that the specimen has, you know, the shape of the base. So if I were collecting these for food, I would have cut off the base, and I would be missing very, very important information that leads me to the ID of this specimen. Okay, so the label is in all the photographs, sections, one side, the other. If there are very big differences in size between what you've collected, place the smaller ones, the larger ones. Make sure you show, you know, the diversity of sizes or even color from your collection. Many times you can't get the label in focus to get the edge of a large mushroom in focus, but then move your focus and make sure you have one blurred label and then one blurred mushroom, but it's the same frame, the same shot. And make sure in your technical photography to capture any detail that may be curious. So for example, in this case, in this Amanita muscaria, it's been eaten probably by a snail or a slug, but there are clear bite marks. And I want to make sure that I get that also in my shot, okay? Here's another case where you see very clearly, you can move the ruler around, you can move the label around, but here in this photograph, I see the top of the cap, I see the side, you know, I see the margin, and I see an open section of the lamellae, the stipe, you know, the base of the stipe, and the details of the flesh. Okay, sometimes they are tiny and you only have one, but you know it's something different and special. So you go through the same steps, but first the entire mushroom, put it on the side, and then finally you cut to get the details of underneath the cap. Okay, and then in that case, and then we move on to a spore print. The spore print is pretty straightforward. Most of you probably know how to do it. We have a mushroom. We cut the stipe off, and we place the cap of the mushroom on a piece of paper, hopefully half white, half black, because if it's on a white piece of paper and it has white spores, you won't be able to see them. And if it's black spored on black paper, you won't see them either. When you do this, the only reason to take a spore print is to be able to use field guides that classify their specimens, first of all, from spore color. I recommend for people that are starting in mycology to do spore prints, but later on, you can use a hand lens and you can see the spores many times deposited either on the annulus, so the ring, or on straight, you can even touch the lamellae, the gills, and you'll get spores on your finger. In the case you do a spore print, keep the stipe and keep the cap afterwards, and always put the label with the specimen, even next to the spore print, okay? And there they are. This is exactly what I was saying. So the reason we use half black or half white paper is pretty clear here, no explanation needed, but it's hard to see white spores on white paper. But not only mushrooms with lamellae, so cap and stipe mushrooms release spores. Gelatinous fungi also release spores. So, before I would, for small specimens, take spore prints directly on a slide, on a microscope slide, because it was easier, they're small, but you can see that these jellies leave a white spore print. And if you have trouble with space or humidity of the paper, you can always take spore prints directly on a glass slide and then keep them. Spore prints are possible from different types of fungal spore producing bodies, or fruit bodies as some people call them. But all of them have spores. So in the case of the cup fungus, many of us, have fun, blowing them or clapping next to them so that they'll release all their spores. You don't want to do that if you're collecting for science. You don't want to have a sporeless specimen. We need the spores for microscopy. So you have to collect them delicately and try to put them in something that's closed so that when they sporulate, you can see the spores, in the tackle box section or something else. In the case of the puffballs, there's no need to take a spore print of a puffball. I mean, it's pretty clear the color of the spores there, but you do want to make sure when collecting puffballs that they are mature or semi-mature, because if you get a very, very new, young puffball and collect it, it won't mature enough to produce spores even. And it's desirable for there to be spores in your specimen to be able to look at them in the microscope. Coral fungi also release spores like the jelly fungus before. You can place them on a piece of paper and the next day you'll have a silhouette in spores. And of course, earth stars are like puffballs. You can just puff a bit out and see what color the spores are. So this is what a typical, rented cabin in the field looks like. You use everything you've got to cover up the specimen so that the wind doesn't blow the spores around. If you can tell, the label is with every single one. I can't stress enough how important it is to keep that label with its specimen, because really later they all look the same in many cases and you won't know which one is which, and all your work is lost. So overnight, leave them covered up. Sometimes, the ones in the middle there weren't enough mugs or glasses, you know, use whatever you can to cover them up so that they're not, so that the spore print is deposited, you know, well deposited on the paper. And then comes probably the most important step, which is drying and storing. So depending on where you're foraying is whether you have one of these or not, right? The infamous food dehydrator that is ideal for drying specimens, okay? Not everybody, certainly not myself, forays where there's electricity. So you won't be dragging this around if you're only going to stop, you know, in a cabin for 2 days, which is the case of this photograph. The rest is dried with silica gel, which are, you know, the silica beads that come in little pouches, sometimes with food or with sneakers, or you can buy it in bulk. And there's a silica that changes color when it's wet, and then you can recharge it. You can dry it out and use it again. Okay, so when you're in, you know, if I'm in deep Patagonia, I would probably carry more silica, but I would definitely not be carrying this around because there's no way to power it. In this case, we stopped in a place with electricity and there was a little heater. So drying, it's important not to cook the mushrooms, please. You have to put them either in a paper bag after the spore print is taken, or after you have all the data, or even the, you know, the wax paper bag, or make a little paper pouch, the label inside with the specimen, and then a duplicate of the label outside, and place them close to a source of heat and air, warm air. I don't know, Ben, if you can help me, but it should not be more than 35 to 40 degrees centigrade is the temperature to dry your specimens. Over 40 degrees centigrade, you degrade the DNA, and it's not ideal.
That's 140 Celsius is 104 Fahrenheit. So that's between around 86 to 100 degrees ish.
Yeah. Thank you very much. Between 84, 86, and 100 degrees Fahrenheit, but not directly on the heat source, with air, with that warm air drying out the specimen. That normally takes, depending on the size of the specimen, it can take anywhere between 1 night to 2 days. But I can't stress how important it is to dry them well. If you don't dry them well and pack them, they will rot. If you don't dry them well and put them, you know, in a Ziploc bag, you know, to keep them, they will rot in there, and you will have lost everything you've done that we've been walking through. So the drying is important. You can use a bit of silica gel and put it inside the bag with a dried specimen, just to make sure that that will absorb extra humidity. The silica will not interact with your specimen unless you're using used silica that may have remnants of another mushroom. Never, you know, if you're going to reuse the silica, you can put the specimen in the bag and you put the bag in the silica. So the silica will not be contaminated with spores or little bits of lamellae or bits of, you know, the cap of the mushroom, but the silica will be absorbing through the paper, okay? And then you can recharge and reuse that silica gel. It's very heavy, and it's not very expensive, but it's something that you want to look after. So we leave them drying overnight, or 1 or 2 nighs. Before you leave the next day to go to the field, you have to, you know, check your specimens. If they're dry, store them, and I'll show how we store them in a bit. And then you start your day. If they're not dry, you can leave the dehydrator on, making sure that it's not more than 100 degrees Fahrenheit, okay? And when they're finally dry, you have a specimen inside a bag with a label that's the same label that's on the field photograph that you took, that's the same label that's on the technical photographs that you took, and then it's the same label that will be on the bag. And ideally, that label should contain this information: the number, the specimen or sample number, who collected it, the person who identified it. So, for example, say Brian Barzee and I are out, in Colorado Springs, and I collect a mushroom, and he says, "Oh, that's Boletus edulis." Then I will write down that he is the person who gave me that name, the location, the coordinates, what it was growing on, and the date it was collected. So above where it says specimen or sample number, you can write a species name and the number, but both things should be on, okay? And this is the final step that will allow you to, I just brought out one box from the fungarium here. I don't know if anybody can see. I'm gonna do this, I hope. Ooh, this smells lovely. But here you can see, for example, hundreds of specimens. Okay, I'm going to pull one out that, let me get the first one so I know where it goes back, that have been collected. Do you see they have?
Giuliana, could you stop this presentation for a minute so we get you full screen again?
Yeah, I'm just going to, I think that was the last slide anyway. So
Perfect.
there it was. So
Perfect.
Here I am. Okay, so here, what you can see in this, so this is one of the boxes of the FFCL fungarium. And we have 2,000 specimens stored. But if you see our specimens, you see the dried fungus. You see the label. That label is the same label, it has the same number as the field photograph. I have a fungarium number, and that will, so you can deposit your specimen later. For example, you can go to the Denver Botanic Garden and go to the fungarium and give Andy Wilson your specimen that's correctly vouchered with the photographs, and he will put it into the fungarium with this other label. And in this case, I make sure they stay dry with the silica gel. Can you see the silica gel, those beads? These beads go blue when wet. Okay, so I know that they're red, I mean my specimen is dry. This is indicating silica, just to make sure that it doesn't rot. The fungus, it has its label, and then it has the fungarium label. And so any one of these that I get out, you'll see the same thing. There's my collecting label, what I've showed you. It has all the information. It has the dried mushroom. This one has indicating silica that has absorbed a bit of humidity, but it's keeping my fungus dry. When you can't separate a sample from, for example, ooh, what was there? Oh, okay, it wasn't me. Sorry. When you can't separate the sample, for example, from a twig, you collect the whole twig, with the fungus attached to it, with its labeling. So sometimes, like in this case, it's a fungus that grows on a leaf, and it's tiny. It's a tiny leaf with the fungus on it. Still collect it, as you see here. It has a label with the number. That's the same number that's in the photograph, in the field photograph, in the technical photographs, in my dried bag, and then in my specimen. So those are basically the absolute fundamentals of collecting for science. I can take any one of these samples to Kew Gardens, to New York Botanic Garden, to Denver Botanic Garden. If I have the photographs and the sample collected the way I've just taught you, your encounter mattered for science. And if you don't do it like that, it's just an anecdote of something special that was photographed, but that was never collected.
Thank you, Giuliana. That's really, really helpful. We had one comment that from James that we should make a paper with these details for reference in the field that we can take with us, because I think it's easy to get excited when you're in the field and starting collecting things, and then you forget to have done the first step.
Exactly, and it happens frequently. It still happens to me, believe it or not, 20 years into this, when I find something really special, I'm bouncing up and down with joy, and suddenly I've cleared the mushroom and I've, to get a good shot, and I'm like, oh, I didn't use a size reference or something. And so it's good to carry a checklist that you will learn very quickly. And in my experience, it's helpful to take the labels pre-made. So, you know, print out if you want with a size bar on it. Sometimes I cut them like little flaps and then I just, you know, rip them off and put them on the specimen. But you hopefully that label will have size reference, a date. If you it doesn't have to have a date, but at least a number, a correlative number that never repeats with another sample. There are some mycologists that start their numbers from 1 every year, but their label has the year attached to it. So it will be, you know, in the case of Ben, so BK01 2020, and maybe you'll get to, you know, BK 200 2020, and then it's 2021 and you'll start with 1 again. I don't recommend doing that. I recommend doing correlative. So now since I've been doing it, I'm close to 1,000. So I have GF, you know, 949. I started late doing that, but it's helped me a lot. It really does, you know, individualize your fungus very, very well.
So a couple years ago, Andy Wilson from Denver Botanic Gardens came and gave us a talk, and he talked about the project that has now been renamed, which maybe you can talk about, but the Colorado Mycoflora project, now renamed to the Fungal Diversity.
Fungal Diversity Survey. Yeah.
Fungal Diversity Survey, which Giuliana was a part of renaming because, of course, Mycoflora, the mushrooms are a different kingdom than plants, right? And so that didn't make sense, right? Or say again.
Or queendom.
Yes, there you go. Is there anything? So Andy Wilson brought, we had some for our club, like little notebooks of printed out, like tear sheets of labels that we can use in the field, which are really helpful, and we can see if we can get some more of those. And then the closest place, of course, for us to take any samples like this would be to the Denver Botanic Gardens. These things, there's a fee associated with submitting samples to the fungarium for DNA sequencing and everything, but that's something that we've been wanting to do more of in our club, and I think this talk really helps us get an understanding of where to begin.
Right, and I would not be surprised that within a year of doing this, somebody from the club will have discovered a new species to science.
And a couple of years ago, James and I found a Amanita or something. It was an Amanita, but we were collecting edibles and there were agaricus everywhere. And I was just cutting agaricus and I picked one and I turned it over and I was like, oh, this is white gills. And then we were like, where's the rest of it? And we started digging around and I popped up the stipe or the vulva and I was able to put it in the bag. Of course, it was cut already, but by the time we got it back to Brian, we couldn't find that anymore. It got lost somewhere in the shuffle and Brian was really upset because we could have found something really interesting and really unique, but we didn't have all the information necessary. So, yeah.
Right. So that illustrates very well what we were talking about, the fact that you need to unearth it carefully, and also the fact that before collecting, you know, take photos, you know, from afar going in. But when you're foraying for food, it's very easy for that to happen. It wasn't your fault. Brian, come on, Brian, don't give him a hard time.
Another thing that Andy Wilson recommended we use, and I only started using it this summer, but it's iNaturalist. It's a really great app. And I think from what you're saying, what you're talking about, it won't really be helpful unless we have the sample, but it's a great way to collect your photos from your phone. It gives you your geo coordinates of your collection. You can add notes in there. I don't know, I could share quickly I could show the website version, not the phone version, but the app is works really well. And I think it's been a really nice way to start. It also really is helpful. Let me share my screen real quick.
You get different people pitching in on the idea as well, which is also very useful. But I think that it's important for the club members to realize that you know, even if you get names from people who really, you know what they're talking about, the voucher, the specimen is irreplaceable.
Yeah. So this is
Wow.
just, this is the website and some things I found from New York, Ohio, and Colorado. But for instance, let's find one. Here's a smooth chanterelle, and you can upload photographs. Okay, and it has a pretty decent, if you have good photographs, which is also key in all of this, right? If you have good photographs, it has a pretty decent kind of, it recommends genus and species. It usually can get genus pretty well if you have a good photograph. And then it says, based on your location, based on the area and other things found in that area, these might be the species. And then what you do is you suggest an ID. So I suggested smooth chanterelle. Here's it has the Latin name and the common name. And then until someone else confirms my ID or suggests another ID and another person agrees with that ID, it is in, it's not in a full, like, it doesn't go to a research grade status, meaning that someone in the community has confirmed your identification. And then, of course, things can be added to collections, other collections, projects. This was automatically added to the lower Lake Erie region project. It has a location tag. If you're collecting edibles and you don't want someone to find your chanterelle spot, you can make it private or you can obscure that location. But if you obscure the location, people can still see that it was found in this area of this state, which is pretty useful but your geodata is there no matter what, which is really useful if you are then later going to take this specimen and get that information and put it into a fungarium. Yeah, but I found it really fun and really helpful. And there are things that sometimes I forget what they are, or I should know what they are. And it's been a way to kind of learn without having a field guide with me, or maybe that I'm in Ohio and my field guide's from Colorado and I just forgot. And it's a really nice way to learn. And it's not just for mushrooms. I'm naturalist. I've been posting photos of insects and plants and things like that. I posted.
Are you from Ohio then?
Say again?
Are you from Ohio?
I am from Ohio, yeah.
Oh yeah, you're proud. I've been in Chillicothe, Ohio.
Yeah, there you go.
My mother lived there for a year when she was a student. There you go.
Really? Yeah. Cool.
Tell a proud Ohioan.
And the moisture, compared to Colorado, there's moisture in Ohio and there's mushrooms in Ohio. More mushrooms. more longer season. But yeah, iNaturalist is a great tool to have on your phone to be using in association with collecting the way that Giuliana has described.
Yes. Yeah, definitely a recommendation. And then if you're, there's another great site called Mushroom Observer that doesn't work on the phone, but there is a great community of professional mycologists and good amateur mycologists on Mushroom Observer, but it's a little more complex to register the data, but it's really, really good. So any of them are useful, but make sure you voucher your encounters because what you see matters.
Thank you. I'm looking, if anyone has any questions, feel free to chat them here. I had one comment that was, someone said, I didn't realize that for when you're collecting edibles, you shouldn't, dig out the mushroom. But my response to that is that, well, if you know exactly what it is, then sure. Right. But if you don't know what it is, you still want to dig underneath and find out what you're looking at.
Well, yeah, and you'll mostly be looking, right. I mean, if you're not certain, you do have to do that. But if there's a beautiful, porcini patch, you could, twist and let it just look. If you twist it, it'll go and it will come out, and then clean right there in the field. But you don't want to, cut out a piece of soil to collect the porcini.
Right.
Yeah.
I don't see any other questions right now. I think this was really clear and great. And the best thing about this is it's going to live on our YouTube page and it's public so other people can see it and we can revisit it in the future. But thank you so much for doing this. And I think next year, if we can do something in person, if we can even do this in person and go through looking for mushrooms and collecting and learning hands-on.
hands-on. I will always be at the service of Pikes Peak Mycological Society. Publicly said.
Amazing. Thank you.
Oh, anything and always, and hopefully next year we can be together not only at Pikes Peak Mycological Society meetings, but also at the Telluride Mushroom Festival.
That's right. And wait, I have a photo of us from last year. Let me see if I can.
Oh, you do?
Yeah.
Ooh. I can't remember what I was. Oh, now I remember. I was wearing a tent last year. Oh, yeah.
from the
What is you?
costume contest. So here's Giuliana, and this is, I don't know which one's me and which one's Jessica and which one is Rita, but we were the Xylaria polymorpha, and there's Giuliana. So we spoke.
I wore a tent that was made out of tent material, which nobody, only I was sweating under it. Nobody realized it was a tent, but that's a great photograph. Thank you very much.
Yeah, thank you. And we have some comments. Just, this was awesome. Thank you, Giuliana. Queendom. Yes, there we go.
Yeah, well,
All right.
I'm not going to say any more.
Thanks again, and we'll see you next year.
Thank you very much. Massive hug and love to all the members, and especially a big hug to you all, to Ben, to Jessica, to Brian, to Mercedes, and James and Mike and everyone.
Awesome. Thanks.
Okay, I would ask them.
Yeah, stay safe there.
Bye from Chile with a lovely condor feather up there. Ciao, ciao.
Ciao.